Hey, it's Jeff Hansen again, and today we're going to look at enzyme kinetics. And in particular, we are going to look very carefully at the Michaelis-Menten equation. I want to apply it to something that's really central to our life, and that is I'm going to look at how our body uses sugar. The most important learning objective in this entire presentation is going to be to understand Michaelis-Menten kinetics. And functionally, what I want you to be able to do is look at a plot of initial enzyme activity versus substrate concentration and to interpret that. Why does it have a hyperbolic shape? What information can you get from it? How can you use that shape to calculate the Michaelis constant Km for a given enzyme? Also, we're going to be applying this to a system of sugar metabolisms, which depends upon glucose transporters 1, 2, 3, and 4. And so I'm going to actually ask you to learn the Km values for them. It's based on some fairly simple physiology. Now, why am I so caught up with glucose? Well, in a week or two, we're going to look at glycolysis. So this is kind of preparing for that. It's a good system. And for that matter, glucose is at the center of life on Earth. Photosynthesis, which makes the fuel that animals ultimately depend upon, is the process of turning carbon dioxide and water into glucose. Half of our dietary energy, humans across the planet, it comes from glucose in the form of starch from plants. Your brain wants to only burn glucose. It will resort to ketone bodies if you're starving to death, but mostly it burns glucose. Your red blood cells exclusively burn glucose. Anaerobic metabolism in muscle uses specifically glucose. In fact, glucose is so important that if you don't have enough glucose in your blood, you die, okay? You really want to keep above 70 or you'll begin feeling miserably. You get down to around 40 or 30, you might go into a coma, and you do that for a while and you cannot survive. Your brain will die. So a very relevant question to you is, how does that glucose get into your brain? You know, the brain has more priority for glucose than any other tissue in your body. But the blood concentration of glucose throughout your body is generally in this range, 70 to 100 milligrams per deciliter. How then is it that the brain can selectively take in more glucose than, say, your muscle or fat? In other words, the concentration of glucose is relatively uniform throughout your body. So how is it that, say, at 80 milligrams per deciliter of glucose, your brain can take in a bunch of sugar, whereas your fat and muscle can't take in as much? And the answer is the brain has a very different type of transporter. There's one type of glucose transporter that brings glucose into the brain at a very low concentration. It brings glucose into muscle and fat at a higher concentration, and it takes glucose into the liver at even a higher concentration. So it is the type of transporters that allow you to differently take into glucose depending on the need of the organ. Now, as long as we're talking about regulation of glucose, I should mention insulin and glucagon, which very powerfully regulate the amount of sugar in your blood, but they do not directly regulate the brain import of sugar. So that if you eat a meal, your sugar immediately skyrockets after the meal, and that signals release of insulin. In turn, as the insulin rises, it signals specifically fat cells and muscle cells to take in glucose at a faster rate, so the glucose starts going down. And as the glucose concentration starts going down, there's less of a signal to take insulin, so its concentration goes down. Opposite of insulin, glucagon has the opposite effect. So when you have a very low glucose concentration, say first thing in the morning, you have quite a bit of glucagon saying, hey, we need more sugar in the blood. But then as you eat, that glucagon concentration plummets as the sugar concentration goes up. But again, insulin and glucagon are important for maintaining the homeostasis of blood sugar, but at a given blood sugar concentration, it is the transporters that are different between the liver, adipose and muscle, and the brain that matter. And let's take a look at this type of transporter. This is through facilitated diffusion. Facilitated doesn't mean that something's putting energy in there to move the glucose. It simply means that glucose is so hydrophilic with its 6 hydroxyl groups that it cannot get through a membrane unless there's some type of opening. So to facilitate random diffusion and not require any energy, there's an opening that glucose can go into or out of. And we can't select which direction it goes other than through concentration. You go from a high concentration gradient on the outside of the cell to a low concentration gradient on the inside, and that's why glucose keeps coming into cells. How does it get trapped? I hope you'll pause the recording and answer that real quick. Hexokinase phosphorylates glucose, and with a phosphate group it cannot get through the transporter, so therefore there's a huge gradient between the amount of glucose on the outside and glucose on the inside. Phosphorylated glucose can't use the transporter, doesn't matter how much of that there is on the inside. Now, in terms of these transporters, it turns out that different types of tissues have different types of transporters. This is absolutely a learning objective. You're going to need to absolutely just outright memorize that the liver has a lot of glucose transporter 2. Adipose and muscle tissue both have a lot of glucose transporter 4. And the brain has a lot of glucose transporter 1 and glucose transporter 3. This is a broadly applicable principle both to pharmacology, physiology, and in fact this is the Michaelis-Menten enzyme kinetics. The most fundamental principle to understand about the Michaelis-Menten enzyme kinetics, and what you absolutely need to know about it to understand anything about enzyme kinetics, is that the Km value represents the concentration of substrate at which the enzyme is half active. Based on that definition, can you answer these 2 questions? I'd like you to pause the video and do it, because if you can pull the answer out of your head, that means you'll know it for upcoming tests, and for that matter, for future lectures. Okay, I'm going to go ahead. I hope you've done it. So an enzyme with a high Km value has a low affinity for substrate. And an enzyme with a low Km value has a high affinity for substrate. So there's an inverse relationship. The way to think about this is if you have a high Km value, that means it takes a high concentration of substrate for it to bind to the enzyme. A low Km value means that at a very low concentration of substrate, you're half bound. So in other words, an enzyme which can bind to something at extremely low concentration would have a low Km value. What is the average concentration of glucose in the blood? Fasting blood glucose. And I think as a physician, you absolutely need to be familiar with that. It's 70 to 100. Let's say it's about 90. I want you to take these values and tell them which of the transporters they apply to, okay? And just look at the tissue and interpret the word. Now I'll give you a clue. Which of these tissues do you think you want to get glucose no matter how low the glucose supply is? And based on that, which of these values will apply there? Number 1, the brain always gets first dibs on everything, right? You need to keep your brain alive. And that means the Km value for GLUT1 and GLUT3 is going to be which of these 3 values? It's going to be 20. Even at 20 milligrams per deciliter, the transporters that bring glucose into the brain are going to be half active. Now, how about the other values? Can you assign them? Let's look at the hepatocytes. Which of the Km values do you think it will have? 20, 90, or 300? And again, I hope you'll stop and think about it. And now I'm going to answer it. We must remember that the hepatocytes are weird cells. They are the altruistic cells. They want to help out everyone else. When you have a massive amount of sugar in the blood, the hepatocytes want to take it in to protect the body from hyperglycemia. When you have an extremely low amount of glucose in the blood, When you're hypoglycemic, then the liver wants to actually produce glucose and release it. Therefore, it wants to have an extremely high Km. What does that mean? It means that when you have a low amount of sugar in the blood, with this high Km value, the transporters on the liver will be nowhere near half bound. They'll be not very active because the liver does not want to take glucose in when the body's starving. It's altruistic. Even at 300 milligrams per deciliter, when you're extremely hyperglycemic, the capacity of these transporters is only half met. So you could even go above 300 milligrams per deciliter, and the import of sugar by the liver will keep going up. And then finally, it sort of makes sense that GLUT4 has a Km value right about a typical fasting blood sugar level. Because if your body's homeostatic at that point, might be nice if sugar going both into fat and muscle, it's only about half active. But it's even a little more complicated than that, and we'll talk about in REM how GLUT4 is the one transporter that is directly regulated by insulin. So basically, when insulin is released, saying you have too much sugar in the blood, then GLUT4 is activated. And when glucagon is released, then GLUT4 is inactivated. And again, we'll talk about that in REM. This 90 milligrams per deciliter is probably a good number to remember a fairly typical blood fasting glucose. I could ask anything about any one of these tissues in terms of how they want glucose and how to interpret these Km values. These Km values will also be very useful when we talk about other enzymes. Before we talk about enzyme kinetics, I want to define some terms. A glucose transporter could be considered an enzyme that converts a glucose on the outside to a glucose on the inside. So that is a change. And in fact, if there's a lot of glucose on the outside, it will more rapidly be converted to glucose on the inside. So considering a transporter to be essentially an enzyme, I now want to look at the rate at which this enzyme catalyzes, so to speak, the movement of sugar from the blood into a cell. So here I have the initial rate at which the sugar is being transported as a function of the sugar's concentration. Of course, this could apply to the rate of any type of chemical reaction based upon the substrate concentration. And one thing you'll notice right away is that as you increase the concentration of substrate, at the beginning there's a big increase in rate. But as you get higher and higher in substrate, that increase in rate begins to taper off, and eventually it approaches an asymptote, a horizontal line that the rate will never exceed, but it will constantly get closer and closer to it. I'd like you right now to tell me, why does it have this shape? In particular, I want to contrast it to the rate of a normal chemical reaction. Normally, if you have a chemical undergoing a chemical change, if you take a certain amount of the substrate, it will go at a certain rate. If you double that amount, it will double the rate. If you double the concentration of substrate again, it'll double the rate again. A linear relationship between substrate concentration and the initial rate of the reaction. Why are enzyme-driven reactions so different? Do attempt to answer it. If you already know the answer, you go ahead. And now I'll explain. An enzymatic catalyzed reaction is first of all going to occur at a much faster rate than a chemical reaction because it's being catalyzed. The energy of the intermediate, so to speak, is going to be lowered to allow it to go from one form to the other. For example, glucose on the outside can go to glucose on the inside in an extremely rapid rate because the transporter is letting it through the membrane, whereas without a transporter, the rate of glucose going through the membrane would be trivially small, imperceptible. Why does the initial rate taper off and approach an asymptote? As you get a lot of substrate, eventually the transporters become completely bound. If every transporter is bound with a substrate, then no matter how much you increase the amount of substrate, it's not going to increase the rate. So in other words, it's enzyme saturation. The idea of enzyme saturation is such an important fundamental concept that I also want to give a visual feel for what the graph is showing. So here, for example, I'm representing a certain amount of glucose, and I'm saying at that concentration of glucose on a given area of the membrane, separating the extracellular space from the cytosol, maybe one glucose transporter will be bound. So let's double the amount of glucose. Now, if there's twice as much glucose extracellularly, probably a couple of these are bound, and that means that they will be going in at a rate twice as fast. Let's say now you dramatically increase the concentration of glucose in the extracellular space. Now, almost every single transporter is bound, and therefore they are topped out. Even if we double the concentration out here again, there aren't any more transporters to bind to. Hence, the hyperbolic shape, enzyme saturation. You never reach Vmax, but it represents the rate at where essentially every transporter is saturated, which you will get closer and closer and closer to, but never reach. So we have the hyperbolic shape. I don't want to go deeply into the Michaelis-Menten equation. However, I would like to compare that equation to the hyperbolic plot. I do think it's worth your while to see that the Michaelis-Menten equation precisely describes this hyperbolic plot. Let's take a look at what happens to VI, the initial rate, as substrate concentration approaches infinity. Will you go ahead and try to do the algebra, take substrate and make it larger and larger and larger, and see if you can algebraically solve the equation? It's really worth your while, but I'm going to go ahead and do it. As substrate gets larger and larger and larger, then the Km value becomes insignificant. So let me explain. Let's say Km is around 10 and substrate is around 10. These are both important terms. But what if substrate goes to 100 or 1,000 or 1,000,000? The bigger substrate gets, the less important Km becomes, and as substrate approaches infinity, Km disappears. It becomes so insignificant it approximates zero. And to be completely clear, Vmax does not disappear like Km because Vmax is being multiplied by the substrate concentration, whereas Km is being added to it. So that relative to substrates, Km completely disappears, but Vmax will stay. So that at infinity, Km would disappear, and you have that Vi equals Vmax times substrate over substrate. And of course, substrate divided by substrate is 1, so that Vi equals Vmax. So what does that mean? The further we go out on the line, the closer we approach Vmax. This is a hyperbolic plot. I'm hoping that makes sense to you, how the Michaelis-Menten gives you this hyperbolic plot, and that the hyperbolic plot simply represents enzyme saturation with substrate. Now let's look at this definition that the Km value is equal to 1/2 the substrate concentration. Why didn't they say that the Km value is at the value at which the enzyme does become saturated? Do you call saturation 90%, 99%, 99.9, 99.99? That value doesn't exist. It turns out by taking half the value, it gives you the powerful algebra and math possible for Michaelis-Menten. Using the definition that the Km value is equal to the substrate concentration at which the enzyme is half occupied, it's half active, can you estimate the Km value from this plot? I want you to go ahead and take a stab at that. It's really worth your while, but I'm going to go ahead. So in order to find the Km value from a simple plot, first you have to look at what the Vmax is. What's the fastest rate that this enzyme can work at? Then you simply go halfway down. The Km by definition is the substrate concentration at which the initial rate is equal to half the Vmax. So you look at the intersect between this horizontal line, halfway between Vmax and the origin. and then you track down. The enzyme is half active when the substrate concentration is about 90 milligrams per deciliter, and I've made it this way because this is going to correspond to the glucose 4 transporter. So that's really all there is to it, and this is probably the simplest way of thinking about Michaelis-Menten kinetics, and you absolutely need to know what a Km value is and some idea how to do simple calculations with it. I now want to talk about how the different Km values for the different cell types automatically regulate their uptake of glucose. For example, let's begin with GLUT4 that's active on fat cells and muscle cells. And let's assume we're at a typical blood fasting level of about 90 milligrams per deciliter, which corresponds to the Km value. Now let's eat a big baked potato or a loaf of bread so that the glucose level in the blood increases dramatically. We've gone up to 300 milligrams per deciliter. How will that affect the rate at which the glucose 4 transporters are acting, even forgetting about the effects of insulin or glucagon? So we're just looking at the cell. There's some glucose 4 transporters. What are they going to do? Well, if the substrate increases, the rate of intake will increase. And this is what's so neat, because even before insulin or glucagon can act, when your blood sugar goes up, automatically the rate of import of sugar into muscle and fat increases quite a bit. And that's really good news because you don't want to be hyperglycemic. You would like instantly for the uptake of sugar to increase. How about if you haven't eaten for 2 or 3 days and so that your blood sugar goes down to maybe 30 milligrams per deciliter? What will happen to the rate at which muscle and fat cells take up glucose? Well, again, there's less there. They're way below the Km value, and so the rate at which they take in sugar will go way down. And this is really good news. Now, think about this a minute. This is automatic, instantaneous regulation of the intake of sugar by muscle cells based upon the fact that the Km value is near the value for fasting blood sugar. And what's really good about this is if you only have 30 milligrams per deciliter of sugar in the blood, you absolutely do not want that sugar going into your muscle or fat cells. You don't want the sugar to go anywhere other than the brain. This was GLUT4 that's in fat and muscle cell. As we mentioned, GLUT1 and GLUT3 in the brain have a much lower Km. In fat and muscle cells, you need to be at 90 before you're half active. So if we're at 30 milligrams per deciliter, the brain is actually more than half active in taking sugar in, whereas the fat and muscle cells are only about a third active. And that's really good news. The brain should get priority. But there's another value to having this extremely low Km value for the brain, so that its transporters are half occupied at only 20 milligrams per deciliter. What happens when you become hyperglycemic? For example, let's say you have 200 milligrams per deciliter. Normally, the brain is going to be taking in sugar at a pretty fast rate. It's almost topped out at a fasting blood sugar level because the brain really has priority. But now we double the concentration, and in fact, the brain's not taking in much more sugar. Why is that good? Well, it's good because the brain not only wants to get sugar under all circumstances, but it wants a constant rate. The brain does not want to be packed full of sugar. You could say that the low Km value of the glucose 1 and 3 transporters ensures that the brain doesn't get too much. It tops out at a very low level, and then it protects the brain from having too much sugar. In fact, the muscle, the fat, and the liver are going to have to deal with hyperglycemia. The job of the brain is to think, not to suck in extra sugar, which could be harmful to it. But let's go a step further, and let's also include the liver in this model. And of course, the liver has a much higher Km value, 300 milligrams per deciliter. And what's nice about the liver is that, again, when we're at fasting blood sugar level, the liver's not taking much in. It's letting the brain and muscle and fat get all that sugar. And for that matter, when we are extremely hypoglycemic, the liver's taking in almost no sugar, which is exactly what you want. You want to ensure the brain gets it. At the far end of the spectrum, the brain is completely topped out at about 100, but the liver, no matter how hyperglycemic you are, 400, 500, it still can go at a faster and faster rate to really get the sugar out of the body, and it has the capability of taking that sugar and making glycogen to protect the body from hyperglycemia. Anyways, I hope you have a good understanding now of how the different Km values allow the different tissues to take in sugar at different rates in order to serve their various functions. That is as far as I want to go today.